Hello. Today we are going to talk about the RT-PCR Lab. It can be a little bit confusing, so we're going to walk through exactly what it is you're doing and what all the reagents you add to the tubes do. Let's start with the big picture. In this lab, we are doing an RT-PCR, or reverse transcriptase polymerase chain reaction. We'll get into what PCR is a little bit later, but let's start with reverse transcriptase first. The first part of our experiment is in the introduction, and it says we will use mouse RNA to make single-stranded cDNA. Now, if you recall from our central dogma discussion, normally in a human we take DNA and transcribe that over into RNA, and then finally we translate it into a protein. That's our normal flow in the cell. However, it might be beneficial to us in some ways if we could actually go back this way. If we could take some RNA that we already have and go back to turn it into DNA, we could then manipulate it or even insert it into another organism. And that is what we're going to do in this lab. We are going to use an enzyme called reverse transcriptase. To make some RNA that we have and turn it back into DNA. Now, something that we should get out of the way right away is if we have RNA, and now I'm talking about mRNA, and we've got a long strand of that, and we know that ends in a whole bunch of A's, because all mRNA has a poly A tail. So, there is our mRNA, the poly A tail, and we know that this is made by copying a DNA strand. However, we also know that originally in the DNA strand, we have a few introns, a few exons, a few more introns, and a few more exons. So maybe we have intron 1, intron 2, exon 1, and exon 2. So this might be the actual DNA strand or of whatever organism we're talking about. And so we cut out the introns, we process these out, and we bring the exons down here. And so, in our mRNA, what we actually end up with are only the exons. So we have exon 1 and exon 2. So, if we were to take this mRNA that's here and already made in an organism and go back up to the DNA, we are only going to be replicating the exons into DNA. We have no idea what the introns are from just this mRNA, because the organism has already cut them out. So we don't know what the introns are, so we're not going to make an exact copy of the original DNA. We're actually going to make an approximation. So, minus the introns, we're going to make what we call cDNA. cDNA doesn't actually exist inside the organism, because inside the organism, the DNA has introns and exons. So that's just kind of an aside about what we're going to do. So we're going to take mouse RNA, that's this messenger RNA here from a mouse, and we're going to make a single-stranded cDNA molecule. Again, this cDNA molecule does not have introns. We don't know what the introns were. They've already been taken out. So we will then use the cDNA as a PCR template and amplify only the GAPDH gene. GAPDH is actually used in glucose metabolism, glycolysis. So it's a very important enzyme, so it's nice that we get to use it. We're going to talk about PCR a little bit later, but for right now, this is basically a DNA copy machine. It makes many, many copies of DNA, so we can do lots of experiments with the DNA we have. So let's go down to step 1. So what we're going to do, we're going to take a 0.2 milliliter microfuge tube, label it, and then we're going to add some things to it. First thing we're going to add is 2 microliters of mouse liver RNA. Now this is total RNA. So what we're going to get is a tube, and it's got all different kinds of RNA in it. It might have messenger RNA, it might have a bunch of tRNAs floating around, which I'll show with a different color. It might even have some ribosome subunits floating around. So we're going to get a tube with a whole bunch of things. However, only one of these types of RNA has a poly-A tail. And that's mRNA. So the mRNA is the only one with a poly-A tail. And we're going to use that to our advantage. So mRNA has a whole bunch of adenines tailing off the end. And what we're going to do is we're going to add some of this oligo D T23 primer. And this is going to bind to the poly-A tail. So now we've got this whole tube full of all these types of tRNA. But we don't want those tRNAs. We actually only want the mRNAs. We're going to add our T23 primer. So what we're going to get in our tube now, we're going to get all of these mRNAs with the poly-A tail, all of these. For simplicity, I'm not going to draw all the other rRNAs and tRNAs, but understand they're still in here. They haven't gone anywhere. Just so my drawing's a little easier to understand, I'm only going to draw the mRNAs. So what we do is we add a whole bunch of these primers, these T23 primers, and they have a whole bunch of T's. And so all of these primers will bind to the A's. So that's what we're going to do in that step. We're also going to add a little bit of nuclease-free water. We don't want nuclease because it will break down the mRNA. And that's not good, obviously. We don't want the mRNAs to break down. So it needs to be special water that does not have any nuclease in it. And that's what we're going to use in our reaction to bring up the volume so everything can work. So now we're going to denature the RNA by incubating it in the PCR machine at 70 degrees. Anytime you see denature, you should remember that from when we talked about enzymes. It means to break down or open up. So what we're going to do, we're going to incubate this at 70 degrees for 5 minutes. Because sometimes what happens with mRNA, because it's single-stranded, it can bend back and actually bond to itself if it has the right chain of nucleotides. Or they can bind to other mRNA strands. But we don't want that to happen. We want all the mRNA strands to be open so they can accept and bond to the T23 primer. So this 70 degrees will break any hydrogen bonds and make sure all the mRNAs are unbonded to anything. And after that, we add a few more things to our tube. We add in our reaction mix, and in this reaction mix, we're going to have things like ATP to provide energy. We're going to have buffer, which is basically special water. We're going to have some nucleotides, so we can make new DNA. These are the things that are going to be contained all in the reaction mix. We're also going to add 2 microliters of reverse transcriptase. If you recall, if we go back up to the top, the very beginning of the lab, we talked about reverse transcriptase polymerase chain reaction. So this is the key player in our lab. This reverse transcriptase, which is over here, is what allows us to go from RNA back to DNA. So remember, we've got a whole bunch of mRNA from the mouse in the tube. We have all the mRNA that the mouse made. It is not just the GAPDH gene that we want right now. So we have not selected for the GAPDH gene yet. It is currently all the mRNA that was from the mouse. And so what we do is to add some of this reverse transcriptase enzyme mix. We're going to add it into our tube. And so we'll draw our tube down here. Same tube. So we're going to have our poly A tails. And of course, we're going to have our poly T primer in green here. And the T's will bind to the A's. And then our reverse transcriptase will be added, and it will run this way. And it will make a DNA copy from the RNA template. So now our tube has some very interesting things in it. The green part is DNA, and the black part on top is still RNA. So now we have these things bonded to each other, and one half is DNA and the other half is RNA, which is kind of cool. Next, we're going to put that mixture inside the PCR machine. And the PCR machine is going to run one cycle. It's going to run 42 degrees Celsius for 1 hour to allow the cDNA synthesis to occur. So this 1 hour in the PCR machine is where this actually happens, where we make the DNA, where the enzyme is going to go along the RNA template and make a cDNA strand. And that will be day 1.